All right, good afternoon, everyone. Welcome to the weekly Living Earth Collaborative and Washington University Evo Pop Bio Seminar Series. My name is Yisan Yang, and I'm one of the LEC postdoc fellows, and I will be hosting the seminar today. So before we get started, because this is new in this semester, I just want to remind everyone again that we are offering the chance for one-on-one meetings with the speakers on the day of their seminars. So our scheduled seminar speaker next week is Dr. Teresa Gildner from Washington University, and she's available to meet with people from 11 a.m. to 1 p.m. next Thursday. So if you want to meet with Dr. Gildner or any future speakers, be on the lookout for the sign-up links in the weekly seminar announcement for available meeting times. So faculty, postdocs, and grad students are all encouraged to sign up to chat with our speakers. And another reminder before we get started, for the LEC fellows and affiliated postdocs, we are hosting a second biodiversity trivia night on Tuesday, September 21st, 7:30 to 9 p.m. So there's still time to sign up. So keep an eye out on the announcement emails that will go out soon. So if you have any questions, please feel free to reach out to Brett Searmore or any of us. So on to introducing our speaker today. It is my pleasure today to introduce Dr. Caleb Axelrod, who will tell us about his research on brain form evolution in freshwater sunfishes. Dr. Axelrod recently joined the Department of Biology at Washington University as a postdoc, and prior to that, Dr. Axelrod got his bachelor's degree in biology at Queen's University in Kingston, Canada. Then he went on to earn his PhD in integrative biology at the University of Guelph in Guelph, Canada. So as you will hear from Dr. Axerod today, his research looks at brain form evolution that are associated with ecological adaptations of different ecotypes in freshwater sunfishes. His work has been published in several influential journals in the field, such as Proceedings of the Royal Society B on Evolution and Development and the Journal of Evolutionary Biology. So before handing off the mic, I just want to remind everyone to leave your comments and questions in a chat box on our YouTube streaming, and Brett and I will relay the questions to Dr. Axelrod at the end of his presentation. So without taking one of his time, take it away, Caleb.
All right, thanks a lot, Yusan, for the invitation and the introduction. I'm really excited to talk to you about the work that I did in primarily focusing on the work that I did during my dissertation. So as biologists, one of the key things we study is why we see so much variation in life. And this shows some of the variation that we can see in animals. Now, as you probably got from my title, I study brains. And this shows some of the variation that exists in brain size among animals. So the top right image there is a sperm whale, which has the largest brain yet identified of any animal. And the top left is a ragworm. The smallest species of which has the smallest brain yet identified in any animal. So the first thing that probably jumps out at you to this, which isn't totally surprising, is that bigger animals have bigger brains. And so one of the other things that we look at when we think about brain size is relative brain size. So brain size relative to body size. And the bottom images show some of that variation. So the bottom right is a tree shrew, which has the largest brain to body size ratio of any mammal. Sorry, it's not humans. And the bottom left is called the bony-eared assfish, which has the smallest brain to body size ratio of any vertebrate. So in terms of why we see such variation, Charles Darwin, our old friend, had some thoughts on this, at least with respect to humans. In The Descent of Man, he wrote that no one, I presume, doubts that the large proportion which the size of man's brain bears to his body, compared to the same proportion in the gorilla or orang, is closely connected with his higher mental powers. Now, I, like many of us, wish we could just take Charles Darwin's lack of doubt as good evidence, but unfortunately, we also need to test whether that variation in relative brain size actually reflects differences in mental powers or cognitive ability, or what that's related to. So I study this question of why we see such variation in brain form using fish. So this is the a side view of a fish brain, of a sunfish brain, the species that I'm going to be talking about. And one thing that might jump out to you initially is that the brain is not homogeneous. It's separated into different regions. And these regions are related to specific functions of cognitive ability. Whole brain size is generally thought to be associated with general cognitive ability, whereas different brain regions are associated with particular cognitive functions that those regions control. And so, for example, the optic tectum in the middle there is the primary visual processing center of the brain. So when I say brain form in my title and for the rest of the talk, I'm referring to both of these characteristics of the brain. So relative brain size, as well as the proportional size of different regions in the brain. The reason that I'm so interested in the brain is because of its links to cognitive ability and cognition. So I define cognition as all information processing done by the central nervous system. And this includes processing of sensory information, motor functions, and higher integrated functions of the brain. This is a really broad definition, but I use it because I want to capture all of the different possible functions that the brain could do, particularly associated with ecology. Now, again, as you might have gotten from my title, the hypothesis that I am going to be working with is that brain form is related to different ecological situations that fish might be living in. So if you think about these two different habitats shown, we can well imagine that it might require different levels of cognitive ability to perform well in these different habitat types. So for example, the habitat on the left might require greater cognitive ability than the habitat on the right because of the more sort of general complex nature of the habitat, requiring greater cognitive ability to survive and navigate in that habitat. You might be wondering why a smaller brain would ever be good. And basically the reason is that the brain tissue is particularly metabolically energetically costly. So it takes a lot of metabolic energy to grow and maintain brain tissue. And so a larger brain gives you greater cognitive ability, but if you don't need greater cognitive ability, then you're just putting energy towards nothing. And so evolving a smaller brain would actually be useful when you don't need greater cognitive ability. I have called this hypothesis that I'm working with the adaptive brain hypothesis, which is that variation in brain form reflects environment-specific functional links between cognitive performance and brain form. And just to create a visual for this, basically this posits that brain form is linked to cognitive ability and behavior, and that cognitive ability is going to be linked to ecological performance in different habitat types. This also, we have to remember, because brain tissue is metabolically costly, that there's always going to be a trade-off between the cognitive benefits of a larger brain and the cognitive and the metabolic cost of a larger brain. And so we have to keep that in mind whenever we're talking about variation in brain form. But generally, this hypothesis predicts that we're going to see an association between brain form and ecology, so environments that differ in their cognitive requirements. There's an issue here, though, which is that this prediction assumes that there are no constraints acting on the ability of brain form to match the particular habitat that it's in. So here I'm defining a constraint as a characteristic of an organism or a population that limits the potential for evolution to drive traits to their phenotypic optimum. Once again, this is a pretty broad definition because I'm trying to capture all of the different types of constraints that might lead to a lack of association between brain form and its environment. And there are many different types of constraints, for example, developmental constraints, evolutionary constraints, or functional constraints. And so this could lead to a lack of association between brain form and the environment when constraints are acting or limiting the ability of brain form to match the environment that it's in. My work in my dissertation and the work that I'm going to be presenting today is framed around three broad questions. So the first is, why does fish form vary independently of body size? The second is change in brain form limited by evolutionary or functional constraint? And finally, what proximate mechanisms contribute to variation in brain form? So I'm going to be presenting a number of different projects that are linked to these particular questions. So getting into the first project I'm going to be talking about. Here I was looking at variation in brain form across a spatial variation of ecological differences, using both intraspecific and interspecific comparisons in order to look at multiple different levels of comparison between these habitats. And this work was published earlier this year in the Journal of Evolutionary Biology, if you want to learn more about it. So I'm going to be looking at, again, variation between types of habitats like the images you see right here. And these habitats are the pelagic and littoral habitats of a lake. So the littoral habitat is the habitat you see on the right there, which is shallow water, near shore, and highly vegetated. The pelagic habitat is in the open water, like on the left, where there's much less structure, though fish in this habitat tend to cluster around shallow rocks. If you've ever been on a boat on a lake and you've had to avoid buoys to try to avoid destroying your motor on a rock, those are the rocks that these fish tend to cluster around. Now, these habitats differ in their ecology. So, for example, in their foraging, fish that live in the littoral habitat tend to forage primarily on larger macroinvertebrates like snails and insect larvae, whereas fish in the pelagic habitat feed primarily on zooplankton. They also differ in their strategies for predator avoidance, as well as just the general three-dimensional structure of the habitat. And they also differ in body morphology. Fish that live in these habitats differ in body morphology, for example, in body form, body shape, as well as jaw morphology. The jaw morphology is going to be important in the second project I talk about, because we use jaw morphology as an indication of foraging ecology, because fish that forage on larger macroinvertebrates, particularly snails, tend to have very wide, robust jaws compared to fish that forage primarily on zooplankton. So the way that I'm looking at these two habitats is using both between species and within species comparisons. So the between species level, the two species that I work with are bluegill sunfish and pumpkinseed sunfish. These species diverged about 15 million years ago, and they differ in their specialization in their ecological specialization. So pumpkinseed are littoral specialists. They are highly specialized to foraging in the littoral habitat, whereas bluegill are pelagic specialists. The key thing about these species, though, is that when they occur in the absence of the other species, so if it's just pumpkinseed or just bluegill in a lake, they will quite often diverge into both habitats. So when pumpkinseed occurs alone, it will move out and colonize the pelagic habitat, leading to ecotypes of the species within each species. And this also occurs in bluegill, where when bluegill occurs alone, it'll move in and colonize both the littoral and the pelagic habitat of the lake. Now, this gives us, the key is that this gives us three different levels of comparison to look at differences in brain form associated with the ecology of the littoral habitat and the pelagic habitat. So first up top, we have a species level comparison based on their historical ecological specialization. And then we have two different within species comparisons. So within pumpkinseed and within bluegill, to look at how these habitats might differ in their cognitive requirements and how that might influence brain form. So in terms of predictions, unconstrained brain form adaptation associated with the adaptive brain hypothesis would predict that we would see differences in brain form associated with ecology across every level of comparison. So in the graphs I'm going to show you here, brain size is on the y-axis and body size is on the x-axis. It might also be brain region size on the y-axis and brain size on the x-axis, depending on which trait we're looking at. And we do this to account for the covariation of brain size, body size, and brain region size and brain size. And in these figures, the yellow or orange lines are going to be showing pumpkinseed species, the pumpkinseed ecotypes, and the blue are going to be showing the bluegill species. And then the lighter colors, so the light yellow and the light blue, are showing the pelagic ecotype within species, and the dark colors, so the darker orange and the dark blue, are showing the littoral ecotype within each species. So in this case, if the littoral habitat is associated with larger brain size, we would expect to see larger brain size in pumpkinseed than bluegill, and larger brain size in the littoral ecotype of both species. So larger brain size in the littoral pumpkinseed than the pelagic pumpkinseed, and in the littoral bluegill than the pelagic bluegill. If enough time has passed that the species level differences no longer matter, we could also see only differences within species. So larger brain size in the littoral ecotypes of both species and smaller brain size in the pelagic ecotypes of both species. And with no differences at the species level, which would still be in line with unconstrained brain form if enough time had passed to remove all of the species level differences. So what we did is we collected individuals from two different lakes. So one that held only pumpkinseed and one that held only bluegill. This is Ashby Lake where I collected the pumpkinseed. And we collected them from the littoral and the pelagic habitats of these lakes. So the arrow here is pointing to a pretty classic pelagic habitat right in the open water. You can't see the rocks from this height of the image, but there are rocks generally in that area that the fish tend to cluster around. And then this is a classic littoral site. So in a shallow bay where there's a much greater sort of shallow habitat for the fish to live in. So we collected them from these habitats and then assessed their body morphology as well as their brain morphology. So we dissected out the brains. This is an image you've seen before. We weighed the brains to get brain size, and then I measured brain region size by taking pictures like this and measuring the length, width, and height of the different regions and calculating the volume of the regions that way. And so what did we find? So first, looking at the between ecotype level. So this is within species between ecotype level comparisons. I found that brain size differs between pumpkinseed ecotypes, but not between bluegill ecotypes. So if you look at this figure, we can see that the littoral pumpkinseed have larger brain size than the pelagic pumpkinseed. So the dark orange line is higher on this graph than the yellow line, but there's no difference between the bluegill ecotypes. And we don't generally see a species level difference because the bluegill both bluegill ecotypes have a similar brain size to the pelagic pumpkinseed. And so we only see a difference in overall brain size here in the pelagic ecotypes. We think being associated with the greater structural complexity in the littoral habitat compared to the pelagic habitat. On the other hand, the telencephalon, which is the region of the brain associated with spatial memory and habitat navigation, that kind of thing, we saw larger telencephalon size in bluegill, pelagic bluegill, and smaller telencephalon size in littoral bluegill, but no difference in telencephalon volume between pumpkinseed ecotypes. Now, the difference in the bluegills could be associated with the particular life history of the bluegill species. So bluegill always nest in the littoral habitat, whereas pumpkinseed nest in both habitats. And so the larger telencephalon size in pelagic bluegill might be because they forage in the pelagic habitat but nest in the littoral habitat. And so they have to have the memory to navigate two different habitats rather than just one. But that's just one idea about why we see that. But generally, the key here is that we are seeing differences in brain form between littoral bluegill ecotypes, I should say, that don't manifest between pumpkinseed ecotypes. And so what we found is that there are differences in brain form between ecotypes in the littoral habitat and the pelagic habitat in both species, but those differences are not consistent, indicating that some aspect of the differences in the life history of these species might lead to different responses in their brain to the habitats that they are colonizing. We also found differences between species that don't manifest at the within species level. So the cerebellum is the region associated with muscular control and body orientation. And that was larger in pumpkinseed than it was in bluegill. But again, no differences between the ecotypes. The optic tectum, which as I said is associated with visual integration, was larger in the bluegill species than the pelagic species, than the pumpkinseed species. But again, we saw no differences between ecotypes. And this demonstrates that there are differences associated with the ecological history of these species between the species that don't manifest at the within species level, suggesting that there are constraints that limit the ability of brain form adaptation in the short period that the ecotypes have been diverging, which is only since the last deglaciation, which would be about 10,000 years, whereas, as I said, the species have diverged for over 15 million years. In terms of conclusions from this section, we found that first, differences in brain form between the species and ecotypes. So consistently across all levels of comparison, we found differences in brain form between the habitats. suggesting that providing general support for the adaptive brain hypothesis. However, inconsistencies in brainform divergence at both the intraspecific and interspecific level suggest that constraints might be important in terms of limiting brainform adaptation to these particular environments. So next, I'm going to get into a project where we were looking at temporal variation in ecology and how that might impact brainform, the link between brainform and ecology. So this work was published last year in Evolution and Development, if you are curious about reading more about this. So this chapter, we're looking at ontogenetic niche shift. And so for a quick background on ontogenetic niche shift, this is the idea that individual ecology changes as individuals age because they get bigger. So there are a number of reasons why we might imagine that ecology might change as individuals' body size changes. So first, in terms of foraging, when you're young and small, you might be way more limited in what you're able to forage on. You can only eat things that are smaller than yourself. So you have a much more specific diet, whereas when you get bigger, you can eat different things and you can eat more things because you're bigger and able to eat bigger things and more difficult things to catch. There might be differences in ecology associated with predation as well. So when you're young, you might be very prone to predation from from larger fish predators, I should say. Whereas when you are older and bigger, you might be much less prone to predation. And so we see these shifts in ecology as individuals age. And from this, the adaptive brain hypothesis would predict that when ecology changes over the course of development, we should see changes in brain size to match those changes in development. To match those changes in ecology as individuals develop. The problem here is that when individuals are developing, their brain is going to be changing, their brain size is changing for reasons completely not associated with ecology, just because the individual is growing and developing. And so the problem is, how do we isolate changes in brain size due to ecology from changes in brain size just due to general development and general brain size changes through life? And the solution that I came up with here was to use two different groups of individuals that diverge in their ecology. So what we need is individuals, two groups that share a niche, share their ecological niche when they're juveniles, but then diverge in their ecological niche as they get older. This can be achieved experimentally, but luckily enough, we had a natural system that showed this type of trend. And the reason this type of shift in ecology is so good for testing this question is because it makes the prediction that we should only see brain size divergence between the groups when we see ecological divergence between the groups. And so what we're looking for is not just how is the brain changing, but we're looking for at what point does the brain start to diverge between the groups, and is that associated with their ecological shifts? So Pumpkinseed, sunfish, one of the species that I work with in the last chapter, is actually a prime candidate for studying this type of question. And the reason is because they share the two ecotypes, the littoral and pelagic ecotypes of Pumpkinseed, share a juvenile niche. They do still live in the two different habitats. So juvenile pelagic Pumpkinseed live in the pelagic habitat, and juvenile littoral Pumpkinseed live in the littoral habitat. But at that point, they don't have any differences in specifically their foraging ecology. They're both just foraging on small small zooplankton prey, because that's the only thing they can forage on. They're not big enough to eat anything else. When they start to age, in the pelagic habitat, they stay very similar in terms of their foraging ecology. They continue to eat the pelagic prey like zooplankton, whereas in the littoral habitat, once they're big enough to take full advantage of the littoral niche, they then diverge and start foraging primarily on those larger macroinvertebrates. So the image on the right there is the image of a very small sunfish that we pulled out of this lake. And so we can make the prediction that brain size, because we already know that there's larger brain size in the littoral ecotype, brain size should only diverge between these ecotypes once they diverge in their ecology. And so we went out to Ashby Lake, where we collected these Pumpkinseed and collected individuals of varying age. So when it says 0-year-old, basically that is the earliest we could collect them, which is once they've reached their juvenile state, their juvenile form, which is about 2 months old is when we collected them there. And then we collected them from every year after that. So we have individuals of 0-year-old, which is again about 2 months, 1-year-old, 2-year-old, 3-year-old, all the way up to about 5 or 6 years old, which is the oldest we were able to collect. And again, we see about an 8% difference in relative brain size between the adults of these fish. Now, I'm going to say now that I'm committing basically a sort of cardinal sin of presentations, and I'm switching up color schemes on you. I promise I'm only doing this once. And so from now on, red is going to indicate littoral fish, and blue is going to indicate pelagic fish, because from now on, we're just using one species. So again, from now on, red is littoral, blue is pelagic. Okay, so the next question becomes, at what age, I should say, do these ecotypes diverge into their adult niches? And so to get at this, we used jaw size, which I indicated earlier, because the jaw size is a good indicator of their foraging ecology. And along with our predictions, so we predicted that jaw size would be the same between the ecotypes when they were younger, and at some point they would diverge once they're able to diverge into their ecological niches. And so what we found is that jaw size didn't differ between the ecotypes until they were 3 years old. So this shows relative oral jaw size on the y-axis and the ages on the x. And at 0, 1, and 2, there was no statistical difference in jaw size between the ecotypes until they were 3 years old. And at 3 years old, they started to show a difference in oral jaw size. At 2, it was close. But generally, this predicts that we shouldn't see any divergence in brain size between these ecotypes until individuals are at least 3 years old, maybe 2 years old, when their ecology begins to diverge. However, what we found is that brain size divergence occurs from the earliest years of life. So in this case, in age 0 fish, we found that while there was no statistical difference in relative brain size, we did see a greater growth rate of brain size by looking at the slope of the line comparing brain size and body size. And so in age 0s, the brain was already growing faster in littoral caught fish. Once individuals were 1 year old, we found larger brain size in the littoral sunfish than in the pelagic sunfish. And this carried through their entire lives. And so we didn't find what we predicted, which is that we would only see brain size divergence once they were 3 years old. And so this indicates that the ecology of adulthood in these fish is more important in shaping development than juvenile ecology, because fish are developing the brain they're going to need as an adult, rather than the brain, and they're developing that from very early life, rather than the brain that is best suited to their environment as a juvenile. And second is that the mismatch between ontogenetic niche shift and brain size development suggests that brain size change is constrained in some way, because if fish could perfectly match their brain to their environment, they probably would. And so there is some constraint limiting the ability to rapidly change the brain in response to changing ecological conditions. So the next project I'm going to talk about is an experiment where we were looking at the proximate mechanisms that contribute to variation in brain form. So 2 different mechanisms that I'm going to talk about here are inherited genetic effects and natural selection, or phenotypic plasticity. So just a quick overview, inherited genetic effects would be that we see match between individuals and their environment based on non-random mortality. So in this red environment, red fish are better suited, so they are more likely to survive than the blue fish, and we end up with an association between the red phenotype and the red habitat. Phenotypic plasticity, and I will say inherited genetic effects occurs at the between generation level. We're talking about evolution there. Phenotypic plasticity is the idea that individuals change phenotype within a single generation to match their environment. So fish start out uncharacterized, the brown, and then they develop into the red phenotype because of cues from their red environment. So to test this, what I did is I took individuals and reared them in conditions that mimic the foraging in the different habitats. So we had the benthic foraging treatment, where individuals were in tanks with a gravel sediment, feeding on large chunks of cube on the bottom of the tank. Whereas the column foraging treatment, meant to mimic the pelagic foraging, they were fed particulate food that floated in the water column. I'll say it was the exact same food, the same substance, it was just given to them in a different form. And so we're really looking at the foraging style as influencing their brain size. And here was the general design of the experiment. So we collected individuals from both the littoral and the pelagic habitat, and then we reared them in both the column and the benthic foraging treatment. And finally, we also included individuals of 3 different age classes, so 0-year-old fish, 1-year-old fish, and 2-year-old fish, to see whether brain plasticity might, the potential for brain plasticity might change as individuals age. In terms of what these different mechanisms would predict, so again, here we're looking at relative brain size on the y-axis, and then we're looking at the 2 treatments on the x-axis, and the source habitat is indicated by the colors of the box plots here. So if we're looking, if the mechanism generating differences in brain size is purely inherited genetic effects, this would predict that we would see differences in brain size between source habitats, but not based on treatment. So we would expect individuals collected from the littoral habitat to have larger brain size than individuals collected from the pelagic habitat, regardless of the treatment they were reared in. Phenotypic plasticity predicts that we will see differences in brain size associated with treatment, but not associated with collection habitat. So we should see larger brain size in the benthic treatment than the column treatment, but no difference in brain size based on collection habitat. Finally, I'm going to talk about another mechanism, which is that we could see evolved differences in phenotypic plasticity, which it means that plasticity itself may have diverged between the ecotypes, in which case we might expect to see plasticity in one ecotype, but not in the other, or a different degree of plasticity. So we could see plasticity in one ecotype, and so see differences in treatment in a certain ecotype, but not in the other ecotype, which would indicate evolved differences in phenotypic plasticity. And in fact, this is exactly what we saw. So in age 0 individuals, I should say these individuals were reared in their treatments for about 6 months and then removed and euthanized. And we looked at their brain size, much as I did in the previous studies. And what we found is that littoral sourced age 0 fish showed no plasticity in response to the foraging treatments. However, pelagic sourced fish did show plasticity. So littoral sourced fish developed large brains regardless of their treatment. Pelagic sourced fish developed large brains similar to the littoral sourced fish in the benthic treatment, but developed smaller brains when they were in the column treatment, indicating that there's been evolved differences in plasticity, and there is greater brain size plasticity in the pelagic sourced fish than in the littoral sourced fish. At age 1, we no longer saw a plastic reaction to in the brain size to the foraging treatments. So there was no difference in brain size between any of the groups that we looked at. And finally, once individuals were 2 years old, we now saw this difference in collection source rather than differences in the treatments. And so we saw larger brain size in littoral source fish and smaller brain size in pelagic source fish, much as we did in the wild collections of the individuals. So apparently, once individuals reach 2 years old, they no longer have the ability, their brains no longer have the ability to respond plastically to their foraging environment. The conclusions from this chapter are, first, is that brain size divergence between these pumpkinseed ecotypes, so the larger brain size and the littoral ecotypes, appears to largely be the result of evolved differences in phenotypic plasticity. So individuals that are born in the littoral habitat develop a large brain regardless of the cue that they receive in their juvenile environment, whereas individuals born in the pelagic habitat develop a smaller brain in response to that pelagic niche. However, if those individuals were transferred to the littoral habitat, we can predict that they would develop a larger, more littoral-like brain size. And second, we see that morphological plasticity in the brain appears to reduce over development. So we see greater plasticity in younger individuals and less plasticity in older individuals. And I'll say that this trend was repeated when we looked at plasticity in jaw size. I am not showing the data here, but we also saw this reduction in plasticity of jaw size as well as brain size. So finally, the final project I'm going to talk about gets back to this question of why fish brain form varies independently of body size. and gets to the question of, does brain size variation, I should say, actually influence ecological performance? So we think it does, based on the association between brain size and different habitats, but I wanted to directly experimentally test the association between brain size and ecological performance. And so we can generally think that foraging on these larger macroinvertebrate prey is going to be more cognitively challenging and therefore require a larger brain size based on our observations from the fish in the wild. So what I did is I took individuals, and they were individuals from the foraging trials, from the experiment that I showed you, and did foraging performance trials with these fish. So the fish were put in tanks, either with Daphnia, so a pelagic prey, or amphipods, scuds, a sort of smallish benthic prey, which scurries along the bottom with a gravel sediment, or again, the pelagic, the Daphnia, was just in the water column. The fish were put into these tanks with the prey, with the live prey, and allowed to forage for 5 minutes, and then transferred to the other tank and allowed to forage for 5 minutes again. We did the treatments in the tests in both directions, so there should be no order effects there. And then we assessed foraging performance by using video analysis and by primarily by opening up their stomachs and counting the number of individuals they were actually able to capture, and then seeing whether there was any association between foraging performance and relative brain size. And this is what we found. So on the right side there, this was success at foraging on Daphnia, so pelagic type foraging. We saw no association between brain size and foraging performance, so indicating that foraging on pelagic prey like Daphnia doesn't present any cognitive challenge. However, with the amphipods, the benthic prey, we saw an association between performance and relative brain size. Individuals with larger brains tended to be more successful foraging on amphipod prey, indicating that foraging on that type of prey is cognitively challenging. And so what we found here is that brain size does indeed influence ecological performance. And we see that prey types differ in their cognitive requirements. So some prey are more cognitively challenging than other prey. And this does align with our expectations that divergence in brain size between habitats is likely related to the different cognitive requirements of foraging in those two different habitats. So in the littoral habitat, they need to forage on more benthic type prey, which is cognitively challenging, resulting in a larger brain size in that habitat. And so this aligns with our expectations of the comparative work that we did. So some general take-home messages from the work that I did in my dissertation. So the first is that brain form appears to be influenced by cognitive requirements of particular habitat conditions, based on comparative and experimental tests of the association between brain form and different ecological conditions, broadly supporting the adaptive brain hypothesis. However, mismatch between ecology and brain form at different stages, so both at the temporal stage and at the spatial scale, looking between and within different species, suggests that brain form adaptation is limited by some form of evolutionary, developmental, or physiological or functional constraint. We also see that brain size divergence in the pumpkinseed, at least, is largely the result of evolved differences in early life brain plasticity between the habitats. And this broadly suggests potentially the importance of the evolution of brain plasticity as an important mechanism generating variation in brain form across species. And finally, brain size influences foraging ecology and foraging performance, but this is different for different types of prey. And so broadly, I did find support that this framework generally works for explaining brain variation in relative brain size and in brain region size, but that we have to take into account differences in mechanisms that can explain this variation, as well as different types of constraints that could limit the match between brain form and ecological performance in different habitat types. So I wanted to end by talking a bit about some of the work that I'm going to be doing in my postdoc at WashU, working with Dr. Swan Gordon and Dr. Bruce Carlson, where I'm going to be looking at brain form variation and looking at testing this general framework using Trinidadian guppies. So for those that don't know, Trinidadian guppies are a really good model system for studying ecological diversification because of their repeated diversification between high and low predation sites in Trinidad. So in Trinidad, guppies live in these inland streams where they occur in streams both above and below waterfalls. So guppies have been able to colonize these habitats above waterfalls, but their predators generally have not been able to colonize the habitats above the waterfall. And so we can get these situations where we have two closely related populations that occur with big differences in the predation regimes, with high and low predation sites occurring close to each other and repeated across different streams. These guppies have been shown to differ in many characteristics of their life history, their morphology, their coloration, their behavior. And so it gives us a really cool opportunity to look at how this ecological variation might be impacting brain variation. So I'm going to be doing a number of projects using this system. And I'm just going to outline two of them for you right now. So the first is starting to get at beyond the morphological variation, getting into some more physiological differences in brain size in terms of how individuals process visual information. So I'm going to be using electrophysiology techniques to measure evoked potentials in the brain of these guppies. So an evoked potential is a electrical response in the brain to a particular external stimulus. And we can use this to start to understand how guppies are processing visual information and how there might be divergence in how guppies from the different habitats are processing visual information. We know that guppies might have different responses to things like predators that sort of across their visual spectrum, as well as different colorations, different colors in males for females doing mate choice. So there are a lot of different characteristics of the guppies that might indicate differences in their ability to process visual information. So I'm going to be using these measurements of evoked potentials in their brains to try to test for differences in how they're processing visual information in their brains. And the second project is I'm going to be trying to test this framework completely. So from start to finish. So I'm going to be looking at brain variation in brain size and brain form, and linking that to cognitive ability and ecological performance. Cognitive ability can be tested using various different types of learning assays. The one I'm going to be using is a spatial learning assay, where we're basically teaching them to navigate a maze and seeing how successful they are at navigating that maze and how long it takes them to navigate that maze. This is a type of characteristics that's been associated with brain size before, but not at the using natural variation and not between individuals of different habitats, like with the high and low predation guppies. Then I'm going to do ecological performance tests, much like the foraging performance tests I described with the sunfish, to see whether we can link brain form variation to cognitive ability, whether cognitive ability is linked to ecological performance on a cognitively challenging prey. And so trying to completely test this framework from start to finish. So with that, I'm going to finish up, and I'd just like to thank my PhD advisors, Baron Robinson and Fred LaBerge, as well as Rob McLaughlin and all the members of the Robinson and McLaughlin labs. The residents of Ashby Lake and Holcomb Lake for providing shelter there and allowing us to collect fish from their lakes. Matt Cornish and Mike Davies from the Hagen Aqua Lab at the University of Guelph, where I did my experiments. And everyone who helped me in the lab and field, and of course the fish for their contribution to my research. Thanks.
All right, thanks Caleb for this awesome seminar. I really enjoyed it, and I look forward to seeing your future work with the guppies. So while we're waiting for the questions to roll in, I will ask one of my own questions that I had. So this one is about the Ontogenetic niche shift project that you had on, I think, the second project. So you're seeing that the jaw is not diverging very early, but the brain is. So do you think that is because there are some other aspects that's like unrelated to the actual feeding itself that is contributing to the difference in development of the brain in these two different habitats?
That's a really good question. And interestingly, initially when we did that project, we were pretty unclear on the mechanism that could be leading to this divergence, because at that point we didn't know whether phenotypic plasticity or inherited genetic differences or what it was that was leading to divergence. But what I think is most likely here is, so when individuals are very, very young, they have this shared ecological niche, which is much more similar to the pelagic adult niche than it is to the littoral adult niche, because they're feeding primarily on zooplankton. And since we now know that littoral sourced pumpkinseed, the juvenile littorals, don't have brain plasticity, they will have the trajectory, their brain development trajectory, to develop a larger brain in that habitat, regardless of the cues they're receiving from their environment. So even though they're receiving pelagic-like foraging cues, they're still developing a large littoral-like brain. In the pelagic habitat, they're receiving these pelagic-like cues as juveniles, but they are developing a smaller pelagic-like brain because they do show plasticity in response to their external cues. So I think that if you were to take a littoral baby and put it into the pelagic habitat, it would still develop a large littoral-like brain, but the reverse isn't true. So if you took a pelagic source fish and moved it into the littoral habitat, it would develop a large, it would develop a littoral-like brain if it was receiving those potential cues, but it still might develop a pelagic brain, I should say, now that I'm rethinking that now, because of the juvenile pelagic cues it's receiving. But I think it has to do more with the differences in plasticity between the ecotypes.
All right, thank you. It's awesome.
Yeah, Caleb, fantastic talk. Thanks so much for giving us this just a really interesting and diverse talk, answering so many different questions in ecology and evolution. I just have, I have a question, then we'll get to Mike's question, but I have a question about your experimental design for the foraging treatments. And, I'm really trying to wrap my head around, there's a lot of different things that are clearly, what do I want to say, driving brain size, right? And one of them, like you showed, is the depth at which they're foraging. But you also said earlier, their habitat in which they're actually like moving around and nesting clearly has an effect. And so my first question here is, how were they housed, right? So did you like have them all housed together and then you would randomly select fish? I'm sorry if I missed this. And do you think if that is the case, how would it have been if you had them housed in separate containers that were relative to their foraging treatments? Do you think you'd have a bigger effect or kind of the same effect? Does this question make sense?
Yeah. So they were housed separately based on their foraging treatment. We couldn't house them together because how do we stop this fish, which is supposed to be in the benthic treatment, from eating the pelagic food that's right next to it? So they were housed separately by tank. So a fish was either in a pelagic type tank or a littoral, a column foraging tank or a benthic foraging tank. They were housed in groups, so replicated groups, which does lead to an interesting question about what is the actual cue that's leading to this difference. We attempted to make the cue about foraging style, so benthic foraging style versus pelagic foraging style, but we have to consider the possibility that there are other things that could be influencing the differences in brain form. One thing that is sociality. So sociality, the interaction between individuals, has been shown to influence brain size and be important for cognitive ability. And that could potentially lead to differences. So even though they were housed in same densities, there might be differences in levels of interaction. So the benthic food was like a chunk of food on the bottom of the tank, which multiple fish could attempt to forage on at the same time. Whereas the column food was particulate food, which might have led to less competition. So it's possible that there were differences in sociality leading to the differences between the treatments. But because the differences that we saw replicated differences that we saw in the field, where there wouldn't be similar sociality differences, we tend to associate it with the foraging style of the different treatments.
Excellent. And just a quick follow-up. So you don't expect to see a difference in the sociality between the two habitats. I would expect that they would be coming more in they would be interacting more with each other in the shallow littoral habitat than the pelagic. Whereas the pelagic, like they're not going to see other fish as much because there's just so much more open space. Is that incorrect?
It's a really interesting question. And actually I would expect the exact opposite. So the pelagic habitat, particularly in Ashby, where we collected the pumpkinseed, had higher productivity than the littoral habitat. And so we actually saw a much higher density of fish out there, sort of clustered around those rocks. I mean, to the point that you could dive down and there would be thousands of fish in front of you in the water. Whereas in the littoral, they're actually a bit more spread out than they are the pelagic. And so both those things, so both of the difference in productivity of the habitat and the difference in sociality would lead us to think, based on those characteristics, that there should be larger brain size in the pelagic if those are what's influencing brain form. But because we saw the opposite, we saw larger brain size in the littoral habitat, we tend to think that those aren't as important for influencing brain size as the foraging styles and navigating the different habitats.
Excellent. Yeah, man, there's a lot going on. Very cool. Thank you.
All right, we have a question from Mike Moore. So interesting talk, Caleb. Do you have any sense of gene flow between the littoral and pelagic ecotypes and the extent to which brain size differences could maintain population boundaries?
It's a really good question. There hasn't been a ton of genetic work done on these ecotypes. What has been done has shown no divergence in neutral alleles between the ecotypes. And so there must be enough gene flow to maintain neutral alleles between the groups, between the ecotypes. We've also done in the lab mark and recapture studies looking at movement between the habitats. And we found that within a summer, there's very little movement between habitats. So within a particular summer, very, very rarely would you see a littoral fish moving out into the pelagic or a pelagic fish moving into the littoral. Occasionally, you'll see it to a slightly greater degree between summers. But generally speaking, we see much more movement between sites within habitats, so between littoral sites or between pelagic sites. And we see very little movement between pelagic and littoral habitats, but there must be enough gene flow to maintain homogeneity of neutral alleles. That being said, because we found differences in their plastic responses, that does indicate that there is at least some divergence in adaptive alleles between these groups.
Cool, thanks.
All right, another question from Bruce Carlson. How can you be sure that a lack of relationship between habitat and brain measures are due to constraints as opposed to other aspects of their ecology not related to foraging? For example, social environment.
So that's a really good question. And I mean, the truth is we can't really. The basis of the constraints idea is that when you see these very similar divergence in ecology, and I'll say I didn't show the data there, but we see the pattern that I showed predicted by unconstrained adaptation is exactly what we found for jaw size. So pumpkinseed had larger jaw size than bluegill, and littoral ecotypes had larger jaw size than the pelagic ecotypes within both species, indicating that our expectation of the differences in their foraging ecology is correct. And since we found that foraging ecology is related to cognitive requirements, both at the experimental level and to some degree at the comparative level, it leads to the expectation that when you have this difference in foraging ecology, you should see a difference in brain size. Now, you're right that could be offset by other characteristics of the environment that don't differ, or potentially that differ in the other direction. So, potentially the reason that we don't see a difference in brain size between bluegill ecotypes is because there's something that's more complex about the pelagic habitat socially, for example, which is leading to larger brain size in the pelagic habitat there than in the littoral habitat, resulting in no difference in brain size. So that is a possibility, but based on the differences that we do see in foraging ecology, we think that the inconsistencies in the difference are due to some form of constraint. But you're right that it could be due to other types of differences in their foraging, in their ecology unrelated to foraging.
All right, this next question is from Benjamin Iberman. Is there any test of the hypothesis that there is any advantage of a smaller brain? So for example, on the isocalorie diet, the larger brain littoral fish are shown to fare worse.
So it's a good question. There's been no test of that in Pumpkinseed or in sunfish. And there's been little to no tests of that actually looking at ecological performance. Generally speaking, what's been done is showing what the trade-offs are of having a larger brain. So what's been called the expensive tissue hypothesis basically posits that when you develop a larger brain, that needs to be offset by reduction in some other characteristics. So the classic one is gut size. So individuals with larger brain size tend to have smaller gut size, potentially smaller gonad size, maybe a shorter lifespan. And so there has been shown that increases in brain size are associated with these decreases in other characteristics of the individuals. Whether that's specifically associated with ecological performance has not yet been tested in fish. So that's something that's that I think would be really interesting to look at.
Thank you.
All right, we have another question from Eric Herzog. He says, thanks, Caleb. At times you suggest that the ecology influences brain size. Do you know how long individuals remain in their littoral or pelagic habitat?
So we know that they spend the entire summer in their habitat because they're foraging and mating out in those habitats. In the winter, we don't know exactly what they're doing because it's very hard to collect them and test what they're doing in the winter. But basically, they're moving to deeper waters, and they aren't foraging very much. They basically just aren't doing much at all during the winter. So when they're foraging and when they're breeding and when they're doing these sort of ecological life requirements, they are living out in those habitats from their first year of life. In terms of what they're doing in the winter and how the winter environment might influence brain form, that's actually a really interesting question and something that I'm hoping to look at in terms of how seasonality might influence brain change over the course of the year.
Awesome. All right, so we're nearing 5 p.m., so I'm gonna wrap up here. So thank you so much again for coming to our seminar, Caleb. And this is a wonderful seminar, and we got some really interesting questions and discussions at the end. And also thanks everyone for tuning in. We will see you next week in our seminar series. All right, bye everyone.
Thanks a lot, Yusan.